Have you ever considered how some diseases, despite, having been around for centuries, continue to defy our expectations, constantly morphing and adapting, presenting entirely new challenges?
It's easy to think we've got a firm grasp on the old ones, isn't it?
Exactly. But the truth, as we'll really try and uncover today, is often far more complex. We're embarking on a mission, a true deep dive into the intricate world of gonococcal infections.
hmm
Our goal is nothing less than to dissect, understand, and truly master this complex subject.
Ah,
We're pulling every single nuance and significant detail directly from Chapter 156 of our comprehensive source material.
that's the plan.
We're talking about gaining the kind of understanding that sets apart elite professionals in this field, a profound grasp of its biology, its impact, and our ongoing battle against it.
That's precisely our objective.
Yeah.
And to achieve that level of mastery, we're going to build a comprehensive, really actionable understanding of Neisseria gonorrhea
Yeah.
and the disease it causes. We'll start with the fundamental definition, then peel back the layers to reveal the intricate microbiology of this organism, explore the dynamic epidemiological trends that shape its spread.
Which are quite alarming recently.
They really are. And we'll unravel the complex mechanisms of its pathogenesis, how it actually causes disease, and interacts with our immune system. Crucially, we'll confront the growing public health crisis of antimicrobial resistance.
A huge topic.
Huge. We'll examine the varied clinical manifestations it presents in real people, detail the precise methods of diagnosis, discuss the very latest treatment strategies, and look at the ongoing efforts in prevention, including, the search for a vaccine. This isn't just about information. It's about gaining that granular knowledge you need as a professional to truly appreciate this public health challenge.
Okay, let's unpack this with the precision an elite professional would demand. We begin at the very core, the definition. Gonorrhea is primarily a sexually transmitted infection, an STI of the epithelium. Now, what that means is it mostly affects the lining, the surface tissues, of various body parts, like the reproductive tract, the rectum, or even the eyes.
Right. It's quite specific in its initial targets.
Think of it as a highly specialized bacterium that targets these specific surface tissues first. And when it first establishes itself, its common primary manifestations, the initial battlegrounds, so to speak, often include cervicitis in the cervix, urethritis in the urethra, proctitis in the rectum, and conjunctivitis in the eyes.
Those are the classic starting points.
These are the points of entry and the first places where the infection takes hold.
And if these initial infections at those epithelial sites go untreated, well, they can lead to some truly severe local complications. And these manifestations, importantly, differ depending on the patient's biological sex.
Okay, so how does that break down?
For female patients, the infection has a concerning tendency to ascend from the cervix upwards. This can lead to endometritis, which is inflammation of the uterine lining, or salpingitis, a serious inflammation of the fallopian tubes.
That sounds bad.
It is. Untreated, it might even progress to a tubovarian abscess, basically a pus-filled mass involving the fallopian tube and ovary. Further complications can include bartholinitis, inflammation of the Bartholin's glands near the vaginal opening, peritonitis, inflammation of the lining of the abdominal cavity, and even perihepatitis.
Inflammation around the liver.
Exactly, sometimes referred to as Fitzhugh-Curtis syndrome. These are significant, potentially fertility-threatening conditions.
Oh. And for male patients.
In male patients, untreated gonorrhea can result in periarthritis, inflammation around the urethra, and epididymitis, which is an inflammation of that coiled tube at the back of the testicle that stores and carries sperm. While less common now with modern treatment, these can be quite painful and impactful. And for newborns, this is critical. The most common and severe local complication is ophthalmia neonatorum, a severe eye infection acquired during passage through the birth canal. If it's untreated, it can tragically lead to blindness. So you can see, even when localized, the potential for harm is significant.
It's precisely this potential for severe consequences that makes understanding gonorrhea like an elite professional so important. While most cases, thankfully, are localized to those initial epithelial sites, there's an uncommon but profoundly significant event known as disseminated gonococcemia, or DGI. This is when the infection manages to break past those local barriers, entering the bloodstream and spreading throughout the body. It becomes a systemic threat.
That's a crucial distinction and a major concern in patient care, definitely. When DGI occurs, its manifestations can be quite varied and, honestly, often perplexing to diagnose if you're not specifically looking for it. You might see characteristic skin lesions, which can be very telling.
Kind of lesion.
They can be papules, pustules, sometimes with hemorrhagic component. Patients might also experience tenosynovitis and inflammation of the tendon sheaths, particularly in the hands and feet, making movement painful.
Right.
And the most common systemic manifestation is arthritis, painful inflammation in the joints. Sometimes it even moves around, migratory.
Okay.
And in rare but very serious cases, DGI can even lead to endocarditis, an infection of the heart valves, or meningitis, inflammation of the membranes surrounding the brain and spinal cord.
So life-threatening potential there.
Absolutely.
Yeah.
These systemic manifestations truly underscore the organism's capability to invade and affect virtually any part of the body, moving beyond the initially localized problem to become a whole-body threat.
To truly master this topic, we need to go deeper, right down to the microscopic level.
Yeah.
Let's shift our focus from the macroscopic symptoms we can see to the microscopic culprit responsible, Neisseria gonorrhoeae itself. For our listeners, how would an expert describe this organism at its core?
Well, to understand how it operates, you first need to know its fundamental nature. Neisseria gonorrhoeae is classified as a gram-negative organism. That's a key characteristic in lab identification. It tells us about its cell wall structure. It's also non-motile, meaning it doesn't move on its own power, and non-spore forming, so it doesn't create resistant spores to survive harsh conditions like some other bacteria do.
Okay.
Under a microscope, you'd typically see it growing singly, as Gonococcal, but its most characteristic appearance, and why it's often referred to as a diplococcus, is that it commonly appears in pairs. Often looks like two coffee beans facing each other, quite distinct.
Right, I've seen those images.
What's truly fascinating and fundamentally important here from a public health perspective is its nature as an exclusively human pathogen.
Meaning only humans get it.
Exactly. This means N-gonorrhea only infects humans. There are no known animal reservoirs. Theoretically, that simplifies control strategies, if not for its other very cunning adaptations. Furthermore, and this is a key insight into its survival strategy, the Gonococcus typically contains, on average, 3 copies of its entire genome per coccal unit.
3 copies. Why?
Well, this phenomenon, known as polyploidy, having multiple copies of its genetic material, permits a remarkably high level of antigenic variation. This means the bacterium can constantly change the proteins on its surface, effectively changing its coat or mass.
Like in disguise.
Precisely. This ability to rapidly alter its appearance and evade immune recognition is absolutely crucial for the organism's survival within the human host. It makes it an incredibly adaptable adversary and a persistent challenge for our immune system and, of course, for vaccine development.
Okay, so it's a shapeshifter at the molecular level.
You could say that. In the lab, like all other Neisseria species, N-gonorrhea is oxidase positive, a specific biochemical characteristic we test for. But what truly distinguishes it from its more benign Neisseria relatives and is vital for its accurate identification in a clinical setting is its unique metabolic fingerprint. It has the ability to grow on selective media, special agar plates designed to inhibit the growth of other competing bacteria that might be present in the sample. And critically, when presented with different sugars, it can metabolize or use glucose for energy, but it conspicuously cannot metabolize maltose, sucrose, or lactose.
Very specific diet.
Very specific. This very precise sugar utilization pattern is one of the definitive laboratory tests used to confirm the identity of Neisseria gonorrhoeae. It's a key piece of the diagnostic puzzle for professionals.
So, understanding the bacterium's basic nature and its immediate impact, what does this all mean for public health on a broader scale? Let's start with the epidemiological trends here in the United States because the numbers tell a surprising and, frankly, quite concerning story.
They really do.
The incidence of gonorrhea had actually been declining steadily for a while, leading some to maybe think we were gaining ground. But then we saw a sharp and alarming reversal. In 2018 alone, there were approximately 616,000 newly reported cases. That represents a staggering 56% increase since just 2015.
A huge jump.
It's not just a bump, it's a significant leap backwards for public health efforts.
Indeed. And to truly grasp the scale of the challenge, you absolutely must broaden that view to a global scale. The World Health Organization estimated that approximately 87 million cases occurred worldwide in 2016.
87 million.
That firmly establishes gonorrhea not just as a national issue, but as a major global public health problem. It's a significant cause of morbidity or illness, particularly in developing countries where resources for diagnosis and treatment are often severely limited.
Okay.
And crucially, it's not just a standalone issue. There's strong and consistent evidence that gonorrhea may play a significant role in enhancing the transmission of HIV.
That connection is critical.
It adds another urgent layer of complexity to its profound public health impact. This synergy is a key reason why global health bodies are so deeply concerned about rising gonorrhea rates.
Delving into the specific demographics predominantly affected here in the U.S., the data from our source shows that gonorrhea disproportionately impacts young, non-white, unmarried, and less educated individuals living in urban populations.
Yes, it highlights existing health disparities.
It really does. It's a disease that, unfortunately, seems to amplify those.
Yes, and for those striving for that elite professional understanding, it's vital to recognize that the number of reported cases we discuss, while alarming in themselves, probably represent only about half of the true number of actual cases.
Only half. Why such a big gap?
Well, this significant discrepancy is due to several critical factors. There's substantial underreporting by individuals or clinics, a common issue with STIs due to stigma or lack of consistent data collection infrastructure. Many people unfortunately engage in self-treatment without seeking a formal diagnosis, or they receive non-specific treatment without laboratory-proven confirmation.
So they might get antibiotics for something else that happens to work.
Possibly, or maybe not effectively. But perhaps most significantly, a large proportion of infected individuals remain completely asymptomatic. They have no symptoms, they don't know they're infected, and they become silent spreaders who contribute continuously to the chain of transmission. That hidden reservoir is a huge challenge.
Let's trace the historical journey of gonorrhea incidence in the U.S. to put this current resurgence into context. We saw reported new cases climb from around 250,000 in the early 1960s to a concerning high of 1.01 million in 1978.
A huge number back then.
The recorded peak incidence in modern times was actually in 1975, hitting 468 new cases per 100,000 population.
Right, and that peak in 75 was attributed to the interaction of several interconnected variables. These included improved accuracy of diagnosis, which meant more cases were simply being counted than before, alongside significant changes in patterns of contraceptive use, specifically a shift away from barrier methods like condoms towards things like the pill, and broader shifts in sexual behavior during that era.
The sexual revolution era?
Exactly. Following that peak, there was a welcome decline in the overall incidence of gonorrhea in the United States over the next quarter century or so. This decline likely reflected increased condom use, which was a significant public health outcome driven by efforts to curtail HIV transmission during that period. It was actually a public health success story we hoped would continue.
But it isn't.
No. As we noted, we're now facing an alarming resurgence. In 2019, the rate was 187.8 new cases per 100,000 population reported in the U.S. That represented a 5% increase in just 1 year and a staggering 92% increase since the historic low in 2009.
92%.
That's incredible.
It is, and this figure is now currently the highest among industrialized countries, a truly concerning position for the U.S. to be in. This alarming trend immediately raises a critical question for professionals. What exactly is driving this resurgence, and what makes it such an urgent threat, particularly here in the U.S.?
Yeah, what's behind it? Our source material makes it unequivocally clear that, simultaneously with this increase in cases, antibiotic resistance is rapidly escalating in the United States and other countries. This dual challenge rising incidence and dwindling treatment options is precisely why the U.S. Centers for Disease Control and Prevention, the CDC, has named antibiotic-resistant gonorrhea as one of the 3 most urgent threats of its kind.
One of the top 3.
Yes, it's not just a health problem, it's a profound public health crisis, because our primary tools for fighting it are steadily losing their effectiveness.
And when we look at the specific groups with the highest attack rates today, who is most affected?
It's predominantly among 15 to 24-year-old women and 20 to 29-year-old men. Over 70% of all reported cases fall within these 2 young age groups.
So young adults primarily.
Yes, indicating a critical need for targeted prevention and screening in young sexually active populations. From an ethnicity standpoint, the rates are highest among African Americans and lowest among persons of Asian descent, reflecting those health disparities again. If we compare the incidence globally, it's notably higher in developing countries than in industrialized nations. However, it's crucial to acknowledge the inherent difficulty in accurately ascertaining the exact incidence of STIs in many developing countries.
Why is that?
Often due to limited surveillance systems, meaning cases aren't consistently tracked, and highly variable diagnostic criteria, leading to inconsistent reporting across regions. Despite these challenges, extremely high rates of gonorrhea have been reported in specific vulnerable populations, such as Aboriginal communities in Namibia and Australia. This highlights pockets of particularly intense public health burden and the need for localized, culturally sensitive interventions.
Here's a fascinating digression our source material brings up, even within this chapter specifically about gonococcal infections. It touches on broader strategies for managing other closely related Neisseria infections, like meningococcal disease.
Right, caused by Neisseria meningitis.
Exactly. This offers us a wider lens, doesn't it? A chance to understand how these bacterial families are tackled by public health and why knowing the specific details of gonorrhea is so vital for professionals in this field.
Absolutely. It provides valuable context. When we're dealing with outbreaks of meningococcal disease, for instance, we distinguish between the primary cases and subsequent ones, which can be either co-primary or secondary.
What's the difference?
Co-primary cases occur soon after the initial case, where transmission is presumed to have originated from the same shared third party. Secondary cases, on the other hand, are direct transmissions from the index case. The risk of secondary cases is highest during the week immediately following the presentation of the index case.
So time is critical.
Very critical. While that risk falls rapidly after the first week, it actually remains above the baseline for up to a year. About 30% of secondary cases occur in the first week, 20% in the second, and most of the remainder crop up within the next 6 weeks. Understanding these timelines is crucial for timely intervention. When it comes to managing these outbreaks, there's a strategic debate between mass prophylaxis and restricted prophylaxis. Mass prophylaxis, where antibiotics are given broadly to a large population, like a whole school,
Right.
has been used in some meningococcal outbreaks. However, the data fully supporting such broad population-level interventions are actually quite limited, and there are significant concerns about widespread adverse events and, critically, the potential for driving the development of antibiotic resistance on a much wider scale across the population.
The unintended consequences.
Exactly. For these reasons, restricted prophylaxis is usually the preferred strategy for meningococcal disease. It's limited to those individuals identified as being at the greatest risk. Specifically, intimate and/or household contacts of the index case, and healthcare workers who have been directly exposed to respiratory secretions. Generally, members of wider communities, like those at schools or colleges where there's been a case, are not offered prophylaxis due to those concerns about broad antibiotic use.
Okay, so if the aim isn't necessarily widespread treatment for the broader community, what's the fundamental goal of this restricted prophylaxis in the context of meningococcal disease for those close contacts?
The primary and precise aim is to eradicate colonization of close contacts with the specific strain that caused the invasive disease in the index case. To maximize effectiveness, prophylaxis should ideally be given to all identified contacts at the same time.
Why simultaneously?
This synchronous administration prevents recolonization by meningococci that might be transmitted from untreated contacts back to those who were treated. It also needs to be administered as soon as possible, not just to eradicate colonization, but also potentially to treat any early undiagnosed disease in secondary cases before it becomes severe. And importantly, if the index patient themselves was treated with an antibiotic that doesn't reliably clear colonization penicillin, for example, they should also be given a prophylactic agent at the end of their treatment course to prevent relapse or onward transmission to others.
Ah, so even the original patient might need it.
Sometimes, yes. It highlights the multi-pronged approach needed for effective bacterial control. When we compare the prophylactic agents used for meningococcal disease, rifampin has been the most widely used and studied. However, it's not always optimal because it fails to eradicate carriage in about 15–20% of cases. There have also been high rates of adverse events reported, compliance issues due to the need for 4 doses over 2 days, and concerning reports of emerging resistance.
Okay, so not perfect. What else?
Ceftriaxone, administered as a single intramuscular or intravenous injection, is highly effective, achieving about a 97% carriage eradication rate. It's a versatile option because it can be used at all ages, including during pregnancy, which is a significant advantage. We have seen occasional reports of reduced susceptibility of isolates to ceftriaxone, which is something we monitor closely given its importance, mirroring our concerns with gonorrhea.
Right.
Ciprofloxacin, or afloxacin, fluoroquinolones, are preferred in some countries. These agents are highly effective and can be administered by mouth, making them convenient. But they're generally not recommended during pregnancy. And of significant concern, resistance to fluoroquinolones has been reported in some meningococci in North America, Europe, and Asia, echoing the resistance challenges we see with gonorrhea.
It seems resistance follows these drugs around.
It certainly does. It's a constant evolutionary battle.
Beyond just antibiotics, our source material also introduces the concept of immunization alongside chemoprophylaxis for documented capsular group A, B, C, Y, or W meningococcal disease. Contacts might be offered immunization with either the Neisseria WY conjugate vaccine or the MenB vaccine as appropriate for the specific strain. This hints at a broader long-term prevention strategy, doesn't it?
Exactly. This combination of immediate antibiotic prophylaxis and longer-term vaccination provides protection that extends well beyond the duration of antibiotic therapy, building a more robust immune defense in the population. And we've seen significant success with mass vaccination efforts in this context.
Outside.
Mass vaccination has been used very effectively to control meningococcal disease during outbreaks, both in closed communities like educational and military establishments, and during epidemics in open communities. This broader perspective on Neisseria prevention strategies is important for an elite professional to grasp because it illustrates how we approach highly adaptable bacteria that can affect public health in multiple ways. It's a theme that resonates strongly with our discussion of gonorrhoea's own challenges. It shows what's possible, and perhaps where gonorrhoea prevention might one day head if we can overcome its unique hurdles. To truly understand gonorrhoea, we need to connect the dots back to its transmission dynamics. If we look at the bigger picture of disease spread, gonorrhoea is transmitted from males to females much more efficiently than in the opposite direction.
Really? How much more efficient?
The rate of transmission to a woman during a single unprotected sexual encounter with an infected man is quite high, approximately 50% to 70%.
Wow, that's very high for a single encounter.
It is. Oropharyngeal gonorrhea, or throat infection, occurs in about 20% of women who practice fellatio with infected partners. Interestingly, transmission in either direction by cunnilingus, from female to female partner, is documented as being quite rare. This efficiency in male-to-female transmission highlights how easily it can perpetuate itself in the population.
Our source material introduces a critical concept for understanding how STIs like gonorrhea persist at a population level. The idea of core group members or high-frequency transmitters. These are essentially a small minority of individuals within any given population who, perhaps due to specific behaviors or circumstances, have very high rates of acquiring new sexual partners.
That's a vital distinction for public health professionals to understand. These core group members are absolutely instrumental in sustaining STI transmission at the population level, acting almost like hubs in a transmission network.
Right.
But another equally instrumental factor in the persistence of gonorrhea in the population is the large number of infected individuals who are either completely asymptomatic or have only minor symptoms that they simply ignore or don't recognize.
The silent spreaders again.
Exactly. Unlike symptomatic individuals who often seek treatment and then, hopefully, cease sexual activity, these asymptomatic persons may not even realize they're infected and therefore continue to transmit the infection unknowingly. This situation fundamentally underscores the importance of proactive public health strategies like contact tracing and empirically treating the sex partners of individuals diagnosed with gonorrhea, even without a confirmed diagnosis in the partner, to break those hidden chains of transmission.
To truly master the nature of this foe, we must go deeper into its cunning strategies. Let's unpack the specific bacterial structures that make gonorrhoea so incredibly adept at causing infection and, crucially, at evading our highly complex immune system. It's truly a master of disguise and deception at the molecular level.
You're right. Its molecular toolkit is incredibly sophisticated. One of the first structures we need to consider are pili, also known as fimbriae. These are filamentous appendages, like tiny, flexible hairs that fresh clinical isolates of gonorrhoea initially express. They allow the bacteria to form distinct colonies that are observable on translucent agar in the lab.
Okay.
But here's a key virulence mechanism and a testament to its adaptability. Pilus expression can be rapidly switched off when the bacteria are subcultured in the lab. This happens due to highly dynamic rearrangements in their pilus genes.
So they can just turn them off.
Effectively, yes. This genetic flexibility is a fundamental basis for the extraordinary antigenic variation of gonococci, allowing them to rapidly change their surface coat, or their identity, and thus evade our host immunity. It's like changing clothes constantly to avoid being recognized. In terms of their direct role in infection, pileated strains adhere much, much better to human mucosal cells, like those lining the reproductive tract, and are significantly more virulent in both organ culture models and human inoculation experiments compared to their non-pileated variants.
So the pillies are key for sticking.
Absolutely key for that initial attachment. In a fallopian tube explant model, for example, pili mediate the initial gonococcal attachment to non-ciliated columnar epithelial cells. This attachment is the critical first step, initiating a cascade of events. Gonococcal adherence, followed by invasion into and then transport through these host cells to intercellular spaces near the basement membrane, or even directly into the subepithelial tissue. This allows the infection to truly take hold and establish itself deeper in the tissue.
And they have another function too.
Yes, crucially, piles are also essential for the process of genetic competence and transformation in gonorrhea. This allows for the horizontal transfer of genetic material between different gonococcal lineages in vivo within the body.
Swapping genes with neighbors.
Essentially, yes.
Yeah.
This is a significant factor in how the bacteria acquire new traits, including the ever-concerning capability of antibiotic resistance.
So it's like a chameleon that can also swap out its internal blueprints. Amazing. Another critical gonococcal surface protein is called OPA, which stands for opacity-associated protein, formerly known as protein 2. What exactly is its specialized role in this pathogenesis and immune evasion?
OPA is indeed very important for both bacterial adhesion and immune interaction. It contributes to what's called intergonococcal adhesion, which is responsible for the opaque appearance of gonococcal colonies on translucent agar when grown in the lab.
Okay, so that's where the name comes from.
Right. Beyond that, OPA directly mediates the organism's adherence to a wide variety of eukaryotic cells, our own human cells, including crucial immune cells called polymorphonuclear leukocytes, or PMNs. Certain OPA variants are particularly adept at promoting the invasion of epithelial cells. This effect has been precisely linked to OPA's ability to bind to various host molecules, such as vitronectin, heparin sulfate proteoglycans, and several members of the carcinoembryonic antigen-related cell adhesion molecule, or CACAM receptor family.
CACAM receptors, those are on our cells.
Yes, these are specific receptors on our cell that the bacteria have learned to exploit. Now, here's a truly sophisticated immune evasion strategy by the gonococcus, demonstrating its mastery of manipulating host biology. When gonococci bind to epithelial CACAM receptors, they can actually prevent the normal shedding or exfoliation of our epithelial cells.
How does that help the bacteria?
By preventing shedding, it helps them stay attached and avoid being cleared away. This happens through a complex mechanism involving nitric oxide, produced during the bacteria's anaerobic metabolism, and the upregulation of a protein called CD105. This interference with our natural cellular turnover may directly hinder the body's natural bacterial clearance mechanisms, allowing the infection to persist longer. Furthermore, specific N-gonorrhea OPA proteins that bind to CECAM1, which is expressed by our primary CD4 plus T lymphocytes, the command and control cells of our adaptive immune system, can actually suppress the activation and proliferation of these crucial immune cells.
So it dampens our immune response.
Exactly, weakening our adaptive immune response right where it needs to be strong. Conversely, some select OPA proteins can engage CEACAM3, a different receptor expressed on neutrophils. This interaction leads to what's called non-opsonic phagocytosis. That is, the neutrophil engulfs and kills the bacteria without needing antibodies or complement to mark it first.
So sometimes it promotes being eaten, sometimes it suppresses immunity.
It's a very nuanced, context-dependent interaction. It allows the bacteria to either evade immune cells or exploit them for entry, depending on the specific OPA variant it happens to be expressing at that moment. It's incredibly adaptable.
It really is like the bacteria has a switch for its interactions. The most abundant gonococcal surface protein is porin, previously designated protein O. Given its sheer abundance on the surface, what's its importance in how the bacteria causes disease and interacts with its human host?
Porin molecules are absolutely fundamental to the bacterium's survival and its virulence, its ability to cause disease. They exist as trimers, groups of 3, forming essential anion-transporting aqueous channels through the bacterium's outer membrane. Think of them as regulated gates controlling what small molecules get into and out of the bacterial cell. Porin also exhibits stable interstrain antigenic variation. This means it can change its surface structure in predictable ways across different bacterial strains. But these changes are stable within a strain type. This characteristic is so consistent that it forms the very basis for gonococcal serotyping, a way labs use to classify different strains based on their specific porin type.
And do these types matter clinically?
They certainly seem to. 2 main serotypes have been identified based on porin. Porladoit 1A strains are often associated with disseminated gonococcal infection, or DGI. meaning they're significantly more likely to spread systemically throughout the body.
The ones that cause arthritis and skin lesions.
Exactly. In stark contrast, poured by 1B strains typically cause local genital infections only. A key characteristic of these DGI strains, the poured by 1A types, is that they are generally resistant to the killing action of normal human serum. Our blood's natural defenses struggle to eliminate them.
So they survive in the bloodstream better.
Precisely. And importantly, they often do not incite a significant local inflammatory response at genital sites, probably because they generate limited chemotactic factors, which are signals that call in our immune cells. This often translates to asymptomatic genital infections, allowing the bacteria to remain undetected locally and thus spread systemically more easily. It's a very clever evasive maneuver.
Wow.
Beyond just being a channel, porin can actively translocate, meaning move itself, to the cytoplasmic membrane of our host cells. This process could potentially initiate gonococcal endocytosis and invasion into our cells, making it an active participant in cellular entry, not just a passive structure. Furthermore, Porbay 1b, when present in outer membrane vesicles, little blebs shed from the bacterial surface during growth, has been shown to inhibit the ability of our dendritic cells to induce T-cell proliferation.
Dendritic cells are key for starting the immune response, right?
Absolutely, they're crucial antigen-presenting cells. By inhibiting their ability to activate T-cells, porin contributes to the gonococcus's ability to subvert our adaptive immunity, making it harder for our body to mount effective long-term defense against reinfection.
It truly sounds like a highly evolved system of evasion and infiltration. But you mentioned there's a whole array of other proteins at work on the bacterial surface too, each with specific functions aiding the bacteria's survival and infection. What are some of these other key players professionals should know about?
Indeed, the bacterial surface is a complex array of tools designed for survival in the human body. Other notable outer membrane proteins include H.8. This is a lipoprotein that's present in high concentration on the surface of all gonococcal strains. This universal presence makes H.8 an excellent target for antibody-based diagnostic testing, allowing us to detect the presence of the bacteria reliably. Then we have a crucial suite of iron-scavenging proteins. Iron is essential for bacterial life, but it's tightly held by our bodies. So, Gonococci have evolved specific proteins to steal it: transferrin-binding proteins, TBP1 and TBP2, lactoferrin-binding proteins, LBPA and LBPB, and haptoglobin-binding proteins, HPUA and HPB. These are all absolutely essential for the gonococcus to scavenge iron from our transferrin, lactoferrin, and heme, respectively, in vivo. Without these specialized proteins, the bacteria would starve for iron and struggle to survive. Interestingly, the presence of transferrin iron has actually been shown to enhance the attachment of iron-deprived gonorrhoeae to human endometrial cells, indicating a direct link between nutrient acquisition and the ability to colonize.
So getting iron helps them stick better too.
It seems so. The gonococcus also has specialized zinc-scavenging proteins, specifically TDFH and TDFJ, which enable them to obtain host zinc from our own proteins like calprotectin and S100 calcium-binding protein A7, also known as psoriasin. This again highlights its dependency on and remarkable ability to exploit host resources for essential metals.
Stealing zinc too.
And finally, there's IgA1 protease. This is an enzyme produced by gonorrhoeae that specifically cleaves or breaks down our mucosal IgA, specifically IgA1. Mucosal IgA is a key antibody component of our immune defenses located in mucous membranes, like in a genital tract. By destroying it, the protease may protect the organism from being neutralized by this first line of immune defense. So when you look at all these proteins together, pili for sticking and variation, OPA for adhesion and immune modulation, porin for transport and immune subversion, plus all these nutrient scavengers and protease, it's clear we're dealing with a very, very well-equipped pathogen, specifically designed for survival and evasion within the human host.
With that incredible arsenal in mind, let's talk about LOS, the lipooligosaccharide. How does this crucial component, part of the bacterium's outer coating, contribute to the infection process and the bacteria's ability to evade our immune system? You mentioned it's different from the LPS and other bacteria.
Yes. Gonococcal lipooligosaccharide, or LOS, is structurally distinct from the lipopolysaccharide, LPS, found in many other gram-negative bacteria like E. coli. It consists of the lipid A component, which is the toxic part, and a core oligosaccharide, but it notably lacks the repeating O-carbohydrate antigenic side chain that's typical of full LPS. That long chain is often what our immune system targets in other bacteria.
So the lack of that chain is important.
It's very significant. Biologically, LOS possesses marked endotoxic activity, meaning it has powerful toxic properties similar to classic endotoxins, capable of triggering strong inflammatory responses. This contributes significantly to the local cytotoxic effect observed, for example, in a fallopian tube model of infection, where it can directly damage host cells and cause inflammation. But a critical and highly sophisticated feature of LOS is its high degree of phase variation in its core sugars. This means the specific carbohydrate structure of LOS can change frequently and dramatically under different growth conditions, even within a single infection.
More shape-shifting.
Exactly. This variation reflects the precise genetic regulation and expression of various glycotransferase genes, the enzymes that dictate how these carbohydrate structures are built. These rapid phenotypic changes have profound implications for its interaction with the host. They can significantly affect how Gonorrhoeae interacts with elements of our humoral immune system, including antibodies and complement proteins. They can also influence the direct binding of the organisms to both professional phagocytes, like macrophages, and non-professional phagocytes, such as our own epithelial cells.
Can you give some specific examples of how this variation helps the bacteria?
Certainly. For instance, Gonococcal that have undergone sialation at their LOS sites, meaning they've added sialic acid residues, a sugar common on our own cells, to their LOS can inhibit the classic pathway of complement activation by reducing the binding of IgG antibodies. This makes them harder for our antibodies to target effectively. They can also bind to complement factor H, a protein in our blood that normally protects our own cells from complement attack. By binding factor H, they effectively inhibit the alternative pathway of complement as well.
So they cloak themselves to avoid complement killing.
Precisely. Both of these mechanisms allow the bacteria to evade being killed by our immune system's powerful complement cascade. Furthermore, LOS sialation may also decrease the non-opsonic OPA-mediated association with neutrophils and inhibit the oxidative burst in PMNs, further impairing the ability of these key immune cells to effectively destroy the bacteria.
But sometimes being unsilated helps them.
Yes, it's context dependent. Conversely, the binding of the unsilated terminal lactosamine residue of LOS to an asialoglycoprotein receptor found on male epithelial cells actually facilitates adherence and subsequent gonococcal invasion of these cells. This highlights how a seemingly simple structural change, adding or removing one sugar group, can dramatically alter host interaction and virulence. Moreover, the oligosaccharide structures within LOS can actively modulate host immune responses in a truly sophisticated way. For example, the specific terminal monosaccharide expressed by the LOS determined which C-type lectin receptor on dendritic cells, our immune system's crucial sentinel cells, is targeted by the bacteria.
And why does that matter?
Because in turn, the specific C-type lectin receptor that's engaged directly influences whether our body elicits a T helper 1, Th1, or T helper 2, Th2 type immune response. A Th2 response may actually be less favorable for the efficient clearance of gonococcal infection. This indicates a very clever bacterial strategy to manipulate our immune system towards a response that allows the bacteria to persist, potentially leading to chronic or recurrent infections. It's really manipulating the immune system's decision-making process.
It's clear that it's not just about the bacteria and its clever tricks. Our own host factors and the nuances of our immune responses also play a significant role in determining how an infection progresses and whether it's effectively cleared. It's a two-way street.
Absolutely. Beyond the direct interactions between gonococcal structures and our epithelial cells, host factors appear to be crucial in mediating the entry of gonococci into non-phagocytic cells. Specifically, Gonorrhoeae has evolved mechanisms to activate certain enzymes within our host cells. Phosphatidylcholine-specific phospholipase C and acidic sphingomyelinase.
What do those do?
Their activation by the bacteria results in the release of diacylglycerol and ceramide within the host cell. And this release is actually a prerequisite, a requirement for the entry of Gonorrhoeae into epithelial cells.
So the bacteria makes our cells help it get inside.
In essence, yes. And the consequences of this are quite severe for the host cell. Ceramide accumulation within the cells can lead to apoptosis or programmed cell death. This programmed cell death may directly disrupt the integrity of the epithelial barrier, essentially creating openings. This, in turn, facilitates the entry of gonococca into the subepithelial tissue, allowing the infection to spread deeper. Additionally, the activation of complement, a key part of our innate immune system, leads to the release of chemotactic factors that draw more immune cells to the site, contributing to inflammation, as does the toxic effect of LS itself, which provokes the release of inflammatory cytokines like TNF-alpha.
What's truly fascinating here and critical for a professional understanding is how specific deficiencies in our own immune system can make certain individuals far more vulnerable to severe forms of these Neisseria infections. It demonstrates a crucial interplay between host and pathogen susceptibility.
That's a perfect point for clinical professionals, absolutely. The critical importance of humoral immunity, our antibody-mediated defenses in protecting us against Neisseria infections, is best illustrated by a specific clinical observation. Persons who are deficient in terminal components, specifically C5 through C9, are profoundly predisposed to recurrent bacteremic gonococcal infections.
The ones that cause DGI.
Exactly. These are individuals who repeatedly experience the systemic spread of gonorrhea into their bloodstream. They're also prone to recurrent meningococcal meningitis, or meningococemia, which are equally serious infections caused by the related bacterium Neisseria meningitidis. This tells us precisely how crucial these specific complement pathways, the membrane attack complex, are in containing Neisseria spread right from the beginning. Beyond complement, our T cells also play a role, although it seems to be a complex and perhaps not always protective one. Gonococcal corin, which we discussed earlier, induces robust T cell proliferative responses in individuals with urogenital gonococcal disease. We see a significant increase in porin-specific interleukin-4, IL-4, producing CD4+, as well as CD8 plus T lymphocytes in infected individuals.
So our T cells are reacting.
They're reacting, which indicates that our adaptive immune system is indeed attempting to mount a specific response to this bacterial component. However, there's a puzzling and really challenging aspect regarding protective immunity from previous infection. Few clear data definitively indicate that a previous gonococcal infection leads to strong, long-lasting protective immunity. This is a critical hurdle for vaccine development. While bactericidal and opsonophosphatic antibodies to porin and LOS may offer some partial protection, it's rarely complete or durable.
So you can get it again easily.
Very easily, unfortunately. And on the other hand, there's a fascinating and rather counterintuitive finding related to another outer membrane protein called RMP, or reduction modifiable protein, formerly protein 3. Our source details that women who become infected and then acquire high levels of antibody to RMP may actually be especially likely to become reinfected with Gonorrhoea.
Wait, antibodies make them more likely to get reinfected? How?
It seems so. The hypothesis is that RMP antibodies actually block the protective effect of other bactericidal antibodies that target porin and LOS. Since RMP itself shows very little, if any, interstrain antigenic variation, meaning it looks pretty much the same on all Gonococci, these RMP antibodies potentially may block antibody-mediated killing of all Gonococci, making the person broadly susceptible to reinfection.
That's incredible, blocking antibodies.
Exactly. The precise mechanism of this blocking hasn't been fully characterized, but it's hypothesized that RMP antibodies may non-competitively inhibit the binding of the protective porin and LOS antibodies, simply because of the close physical proximity of these structures in the gonococcal outer membrane. This sophisticated and detrimental interplay can significantly influence the outcome of experimental challenge with gonorrhea. To truly connect this phenomenon to broader microbial exposure and environmental factors, it's worth noting that RMP's extensive homology, meaning significant structural similarity to enterobacterial Ompir and meningococcal class 4 proteins.
Proteins from other bacteria.
Yes, often harmless bacteria that live in our gut or respiratory tract. This strong homology suggests that these unusual blocking RMP antibodies might actually result from prior exposure to cross-reacting proteins from these other, often commensal, bacterial species.
So exposure to harmless bacteria could make you more susceptible to gonorrhea later.
It's a possibility being investigated. It suggests that even prior exposure to seemingly harmless bacteria could potentially play a role even in first-time infections with gonorrhea by priming the body to produce these blocking antibodies. This highlights the incredibly complex and interconnected interplay between different bacteria and our human immune system, where previous exposures can have unexpected and even detrimental consequences for new infections. This is a deep and challenging area of research, especially for vaccine development, trying to elicit the right kind of antibody response.
That last point about previous exposures and blocking antibodies is truly a profound insight into the complexity of our microbial landscape. But now, this brings us squarely to one of the biggest, most urgent challenges facing public health today. Gonorrhoea's remarkable, almost frustrating capacity to alter its antigenic structure and adapt to changes in its microenvironment. This relentless adaptability has led to widespread resistance to numerous antibiotics, transforming what was once a relatively straightforward infection to treat into a significant, global public health crisis.
Indeed, the history of drug resistance in gonorrhea is quite a sobering chronicle. It really demonstrates bacterial evolution happening in real time, right before our eyes. The first effective agents against gonorrhea were the sulfonamides, introduced back in the 1930s.
Yeah.
But they tragically became ineffective within about a decade due to rapidly emerging resistance, showing just how quickly this bacterium can adapt when pressured. Penicillin was then adopted as the new drug of choice for treatment, a true game changer at the time. But by 1965, even penicillin was showing cracks. A significant 42% of gonococcal isolates had developed low-level resistance to penicillin G. And then a major hurdle emerged with the later development of high-level resistance due to the production of penicillinase, also called galactamase, an enzyme that directly breaks down penicillin, rendering it completely useless against those strains.
So the bacteria fought back with enzymes.
Exactly. This was a critical turning point in treatment strategies. For professionals, understanding how Gonococci become fully resistant to antibiotics is absolutely key. It primarily happens through two main routes, either by chromosomal mutations, which are changes in the bacterium's own core DNA, or by the acquisition of R factors. These are small, circular pieces of DNA called plasmids that can carry resistance genes and, importantly, can be easily transferred between bacteria, sometimes even across different species.
Okay, so changes in their own genes are borrowing genes from others.
Precisely. Within chromosomal mutations, two types have been precisely described. The first type is drug-specific, often a single-step mutation that leads to high-level resistance to a particular drug. The second type involves mutations at several different chromosomal locations, or loci, that combine synergistically to determine both the overall level and the specific pattern of resistance to multiple drugs. Strains with these chromosomal gene mutations were observed as early as the late 1950s, and as recently as 2007, chromosomal mutations still accounted for resistance to penicillin, tetracycline, or both in approximately 16% of strains surveyed in the United States, indicating their persistent role alongside plasmids. The spread of plasmid-mediated resistance has been incredibly rapid and globally impactful. Galactamase, penicillinase, producing strains of N. gonorrhoeae, often referred to as PPNG, which carry these galactamase plasmids, had rapidly disseminated worldwide by the early 1980s. That quickly. What's even more concerning is the phenomenon of co-occurrence. N. gonorrhoeae strains with plasmid-borne tetracycline resistance, or TRNG, have the ability to mobilize or help transfer certain butyllactamase plasmids. This means that PPNG and TRNG often occur together in the same bacterial strain, and sometimes even alongside strains exhibiting chromosomally mediated resistance, known as CMRNG.
So they pile up different resistance mechanisms.
Exactly. This complex web of overlapping resistance mechanisms has rendered penicillin, ampicillin, and tetracycline fundamentally unreliable for the treatment of gonorrhea today. Current professional guidelines strongly advise against their use entirely.
So as penicillin and tetracycline failed, we moved to newer drugs, the quinolones. How did N. gonorrhoeae respond to those, and what new challenges did that create?
Well, quinolone-containing regimens, which included fluoroquinolones like ciprofloxacin, were widely recommended for a time. They even offered the added advantage of antichlamydial activity when administered for a 7-day course, which seemed convenient. However, predictably perhaps, quinolone-resistant N. gonorrhoeae, or QRNG, rapidly emerged soon after these agents were first introduced to treat gonorrhea.
History repeating itself.
It certainly felt like it. This resistance became particularly prevalent in the Pacific Islands, including Hawaii, and throughout Asia, where, in some specific areas, virtually all gonococcal strains are now resistant to quinolones. At present, QRNG is also common in parts of Europe and the Middle East. In the United States, while QRNG has been identified in all areas, it's predominantly seen in states along the Pacific coast, which is where the resistant strains were first observed entering the country. For the expert, the mechanisms implicated in this fluoroquinolone resistance involve specific alterations, mutations, in bacterial enzymes called DNA gyrase and topoisomerase II. These enzymes are critical for the bacterium's DNA replication and maintenance. If these enzymes are altered, the drug cannot bind effectively, and the bacteria simply continue to multiply despite the antibiotic. And it's not just quinolones. Resistance to spectenomycin, which has been used in the past as an alternate agent, especially for penicillin allergic patients, has also been reported. While spectenomycin traditionally wasn't associated with cross-resistance to other antibiotics, making it potentially useful for multi-drug resistant strains, Outbreaks caused by strains resistant only to spectinomycin have been documented in places like Korea and England when the drug was widely used there.
So even the backup options started failing.
Exactly. This highlights a recurring pattern. Widespread use often correlates with the emergence of resistance, forcing us to constantly seek new solutions and protect the ones we still have.
This raises an extremely important and urgent question for any professional in this field. What's considered our last, truly effective line of defense? The third generation cephalosporins, and how exactly are they holding up against this incredibly adaptable bacterium? Are we approaching a dangerous tipping point?
That is precisely the question that keeps public health experts and infectious disease specialists awake at night. Ceftriaxone, a third-generation cephalosporin typically given as an injection, remains the most reliable agent and is currently recommended as the single-dose therapy for uncomplicated gonorrhea. However, there's significant and growing concern due to the recent isolation of strains that are highly resistant to ceftriaxone, with minimal inhibitory concentrations, or MICs, which is the lowest concentration of an antibiotic that prevents visible growth reaching as high as 2 micromolar.
That sounds very high.
It is. These concerning strains have been reported in Japan and some European countries. Now, despite these rising MICs, for example, from 0.0001-0.00 APA-mL for fully susceptible strains up to 0.015-0.125 megamel for strains with reduced susceptibility, the routinely recommended parenteral dose of ceftriaxone, the injection, still achieves drug levels in the blood, the urethra, and the cervix that greatly exceed these MICs.
So the current dose still works, mostly.
Yes, for now the current high dose remains largely effective for uncomplicated infections in most areas, but it's a constant race and the margin for error is shrinking. A crucial decision reflecting this concern was made regarding oral cefixime. This was previously the recommended alternative oral third-generation cephalosporin. But its rising MICs against N. gonorrhoeae worldwide, combined with this drug's limited capacity to reach levels sufficiently higher than those MICs in the blood, the urethra, the cervix, and especially the pharynx, a particularly challenging site for antibiotic penetration, have led to its removal from the list of first-line agents for the treatment of uncomplicated gonorrhea.
So oral options are becoming much more limited.
Very much so. This was a direct response to observed treatment failures with cefixime. For an elite professional, understanding the precise molecular mechanisms behind this reduced susceptibility to ceftriaxone and cefixime, what we call cephalosporin intermediate resistant strains, is paramount. These strains often contain mutations in the pain allele. This is considered the principal resistance determinant, and it encodes a penicillin binding protein, PBP2, whose amino acid sequence can differ by up to 60-70 amino acids from that of the wild-type PBP2 found in susceptible strains.
And PBP2 is the target of the drug.
Exactly. These changes make the PBP2 less able to bind the cephalosporin antibiotic effectively. Other mutations often contribute synergistically as well. Mutations in the multiple transferable resistance regulator, or MTRR, gene can lead to increased drug efflux through the MTRCDE efflux pump. This pump literally pushes the antibiotic out of the bacterial cell before it can reach high enough concentrations to work. And mutations in PIN-B, which codes for the porin protein we discussed, can decrease drug influx into the cell through PORB, essentially reducing the amount of drug that even enters the bacterial cell in the first place.
So they block it from getting in, pump it out if it does get in, and change the target.
Yes. The bacteria often employ multiple sophisticated genetically encoded strategies simultaneously to keep the drug from reaching its target, or to remove it once it gets inside. It really showcases the power of evolutionary pressure. Lastly, azithromycin resistance is also a growing concern. This can result from specific alterations of the ribosomal binding target where azithromycin normally acts, or, similar to cephalosporins, from the over and under expression of efflux and influx systems. What's particularly alarming for public health globally is that combined resistance to both cephalosporins and azithromycin have been reported in several instances throughout the world.
The dreaded super gonorrhea.
That's the term often used. This dual resistance significantly limits our remaining treatment options, creating nightmare scenarios for clinicians and public health officials. It poses a severe threat to our ability to effectively treat gonorrhea and makes the search for new antibiotics and effective prevention strategies like vaccines more urgent than ever before.
That deep dive into resistance truly underscores the urgency. Now, let's shift our focus from the abstract microbiology and the alarming rise of resistance to how these infections actually present themselves in real people, the clinical manifestations. We'll start with how it typically appears in men.
Okay. In male patients, acute urethritis is far and away the most common clinical manifestation of gonorrhea. The usual incubation period after exposure is typically 2 to 7 days, though it can certainly be longer. And critically, a significant proportion of men actually remain completely asymptomatic.
Which drives spread, as we discussed.
Absolutely. This asymptomatic carriage is a major driver of continued transmission, as these men are unaware they are infected and infectious. Interestingly, strains of the PORB.1a serotype, which we linked earlier to DGI, also tend to cause a greater proportion of cases of milder and asymptomatic urethritis compared to PORB.1b strains. This might explain, in part, why some infections destined to disseminate fly under the radar initially.
Makes sense.
When symptoms do occur in men, the major ones are urethral discharge and dysuria, which is painful urination. The discharge initially might be scant and mucoid, but it typically becomes profuse and purulent, or pus-like, within a day or two. It's often described as thick and yellowish green. The dysuria usually occurs without urinary frequency or urgency, which can sometimes help differentiate it clinically from a typical bladder infection or cystitis. A rapid presumptive diagnosis in men can often be obtained by a gram stain of the urethral discharge. As we mentioned, this microscopic examination may reveal polymorphonuclear leukocytes, or PMNs, those inflammatory white blood cells, along with characteristic gram-negative intracellular Gonococcal and diplococcal. Seeing the bacteria inside the PMNs is highly suggestive, as shown in Figure 156-1 in our source material. This quick result can often guide initial empirical treatment while awaiting more definitive tests.
But it's not always straightforward.
No. While the clinical manifestations of gonococcal urethritis are generally more severe and overt than those of non-gonococcal urethritis, including urethritis caused by Chlamydia trachomatis, it's important for professionals to remember that exceptions are common. It's often impossible to definitively differentiate the causes of urethritis based on symptoms alone. In fact, the majority of urethritis cases seen in the United States today are not caused by N. gonorrhoea or C. trachomatis.
Really? What else causes it then?
Well, many of these cases don't have a specific etiologic agent identified, although other organisms like Mycoplasma genitalium or ureaplasma may be responsible. It's also worth mentioning that certain clones of Neisseria meningititis, the other pathogenic member of the Neisseria family, have been documented to cause urethritis outbreaks in men who have sex with men, MSM, in Europe, and also in heterosexual men in the southern and midwestern United States. This further complicates the clinical picture if you're not thinking broadly about potential causes.
So what does this high rate of asymptomatic infection, especially in men, mean for the overall spread and control efforts? And how does it play into the public health challenge we've been discussing?
It means that while most symptomatic men with gonorrhea typically seek treatment and thus hopefully cease to be infectious fairly quickly, the large number of asymptomatic men poses a significant and ongoing challenge for control. These asymptomatic men accumulate in the population over time, and at any given point, they constitute about 2/3 of all infected men. Together with men who are still incubating the organism but are asymptomatic and shedding bacteria, they serve as the primary, often completely silent source of ongoing infection spread in the population. It's a major reason why screening is so important. Before the widespread availability of antibiotics, symptoms of urethritis could persist for approximately 8 weeks, which also contributed to prolonged infectious periods back then. Now, it's really the asymptomatic period that's the key challenge for interrupting transmission. While in the past, before effective treatment, gonorrhea could lead to significant local complications in men, these are now thankfully quite uncommon due to earlier diagnosis and effective antibiotics. For example, epididymitis, that inflammation of the epididymis, is now an infrequent complication. And gonococcal prostatitis, infection of the prostate, occurs rarely, if at all. Other unusual local complications that used to be seen more often include edema of the penis due to dorsal lymphangitis or thrombophlebitis, a submucous inflammatory soft infiltration of the urethral wall, periurethral abscess or fistula formation, inflammation or abscess of Cowper's gland near the urethra, and seminal vesiculitis. Balanitis, inflammation of the glans penis, may also develop in uncircumcised men. Understanding these historical complications helps professionals appreciate the progress made in treatment, but also the continued need for vigilance and prompt management.
Now, what's particularly fascinating and often challenging for professionals is the varied ways gonorrhea can present in women. It's often less obvious or more generalized than the typical male presentation, which can sadly delay diagnosis and treatment, potentially leading to more severe long-term outcomes.
That's a key clinical challenge that truly highlights the importance of thorough screening and maintaining a high index of suspicion in women. Mucopurulent cervicitis is a very common STI diagnosis in American women and can be caused by N. gonorrhoeae, but also C. trachomatis, and other organisms including Mycoplasma genitalium. This cervicitis may also coexist with candidal yeast or trichomonal vaginitis, further complicating the clinical picture and symptoms. N. gonorrhoeae primarily infects the columnar epithelium of the cervical os, which is the opening of the cervix into the vagina. Bartholin's glands, located at the vaginal opening, are also occasionally infected. While most women infected with N. gonorrhoeae eventually develop some symptoms, those who remain completely asymptomatic or have only minor, vague symptoms may delay seeking medical attention, allowing the infection to persist or ascend. These minor symptoms might include scant vaginal discharge originating from the inflamed cervix. It's important to note this is often not due to vaginitis per se, but cervicitis. And dysuria, painful urination, often without the urgency or frequency typically seen in bladder infections. This dysuria might be associated with concurrent gonococcal urethritis. Although the incubation period is less well defined in women than in men, symptoms usually develop within 10 days of infection and tend to be more acute and intense compared to those of chlamydial cervicitis, which can be a subtle clinical clue if present.
And what might you see on examination?
On physical examination, a mucopurulent discharge, a pus-like mucus, may be seen issuing from the cervical os, or the cervix itself might appear reddened and inflamed, friable, even if the patient reports no specific symptoms. It's absolutely crucial to understand, as we mentioned in diagnostics, that Gram stain is not sensitive enough for reliably diagnosing gonorrhea in women due to the normal vaginal and cervical flora. Therefore, specimens should always be submitted for culture or another suitable non-culture diagnostic method like NATs. In contrast, chlamydial infection more often presents with an edematous and friable cervical ectopi and endocervical bleeding upon gentle swabbing, offering a potential differential hint during the exam.
And the risk of upward spread is significant.
Very significant. If the gonococcal infection extends deeper upwards from the cervix, it can cause dyspareunia, which is painful intercourse, and lower abdominal or back pain. In such cases, it is absolutely imperative for the clinician to consider a diagnosis of pelvic inflammatory disease, or PID, and to initiate appropriate treatment for that more serious condition immediately, as discussed in other chapters of our source material. This highlights the dangerous ascending nature of the infection in women if left untreated. It's also worth noting that N. gonorrhoeae may also be recovered from the urethra and rectum of women who have cervicitis, but it's relatively rare for these to be the only infected sites. This occurs in only about 5% of women with gonorrhea. Urethritis in women may produce symptoms of internal dysuria, often mistakenly attributed by the patient to a cystitis or bladder infection. The presence of pyuria white blood cells in the urine, in the absence of significant bacteriuria visible on a Gram stain of unspun urine, and with urine cultures that fail to yield a high bacterial count typically associated with UTI, strongly suggests the possibility of urethritis.
This is most commonly due to C. trachomatis. But urethral infection within gonorrhea can also occur in this context. In the case of gonorrhea, urethral cultures are usually positive if obtained. This clinical picture, pyuria without bacteriuria, requires a sharp clinical eye and appropriate testing for STIs. Now, regarding gonococcal vaginitis, specifically inflammation of the vagina itself, is actually quite rare in healthy estrogenized women.
Why is that?
This is because the vaginal mucosa of healthy women is lined by stratified squamous epithelium, which is generally resistant to infection by N. gonorrhoea. However, gonococcal vaginitis can occur in anestrogenic women, those with very low estrogen levels, such as prepubertal girls and postmenopausal women. In these individuals, the vaginal stratified squamous epithelium is often thinned down significantly, sometimes just to the basilar layer, which can be infected by N. gonorrhoea. This infection typically results in intense inflammation of the vagina, making a physical examination, speculum and bimanual, extremely painful. The vaginal mucosa appears red and edematous, and there's often an abundant purulent discharge present. Infections in the urethra and in Skene's and Bartholin's glands often accompany gonococcal vaginitis in these patients. Inflamed cervical erosion or abscesses and Nabovian cysts may also occur, sometimes with coexisting cervicitis.
Let's connect this now to specific anatomical sites and population groups that face unique challenges, starting with rectal gonorrhea. This is a particularly important area for public health, especially as we've seen it contribute to the problem of antibiotic resistance.
Absolutely. Due to the anatomical proximity and the potential spread of cervical exudate downwards to the rectum, N. gonorrhoea is sometimes recovered from the rectum of women with uncomplicated gonococcal cervicitis. However, as noted previously, the rectum is the sole site of infection in only about 5% of women with gonorrhea. These women are usually asymptomatic, but occasionally they may have symptoms of acute proctitis, which can manifest as anorectal pain or pruritus, itching, tenesmus, a feeling of needing to defecate even when the bowel is empty, purulent rectal discharge, and rectal bleeding.
And what about in men who have sex with men?
Among men who have sex with men, or MSM, the frequency of gonococcal infection, including rectal infection, saw a significant decline of 90% or more throughout the United States in the early 1980s. This was largely attributed to safer sex practices adopted in response to the HIV epidemic. However, there's been a well-documented and concerning resurgence of gonorrhea among MSM in several major cities since the 1990s, with estimated rates of reported cases having more than doubled in a recent 3-year period alone. This highlights a significant reversal in public health trends for this population. A critical finding here, vital for professionals tracking resistance patterns, is that gonococcal isolates recovered from the rectum of MSM tend to be more resistant to antimicrobial agents than gonococcal isolates recovered from other body sites, like the urethra or cervix.
Why is that? Is there a biological reason?
Yes, this increased resistance appears to be genetically linked, at least in part, to mutations in the mTOR gene or in its promoter region. As we touched on earlier, these mutations result in increased drug efflux, meaning the bacteria actively pump out certain antimicrobial hydrophobic agents. This includes things like bile acids and fatty acids that are naturally present in high concentrations in feces. This constant exposure and the ability to pump out these substances likely selects for strains with higher baseline resistance, which may have contributed significantly to the higher rates of treatment failure observed for rectal gonorrhea when older regimens, such as penicillin or tetracyclines, were used. It really underscores how the specific infection site, the local microenvironment, can profoundly impact both resistance development and subsequent treatment efficacy.
What about infections in the throat, known as pharyngeal gonorrhea? This is another site with unique diagnostic challenges and important implications for resistance development.
Pharyngeal gonorrhea is usually quite mild or even entirely asymptomatic, which makes it hard to detect without specific screening. Though symptomatic pharyngitis, a sore throat, does occasionally occur, sometimes it's accompanied by cervical lymphadenitis, which is swelling of the lymph nodes in the neck. The infection is acquired through oral genital sexual exposure, with fellatio being a more efficient means of transmission than cunnilingus. In certain female adolescent populations in the United States, pharyngeal gonorrhea has actually become as common as genital gonorrhea, highlighting its prevalence in some groups.
Does it spread easily from the throat?
Thankfully, most cases of pharyngeal gonorrhea resolve spontaneously over time, and transmission from the pharynx to sexual contacts seems to be rare. However, pharyngeal infection almost always coexists with a genital infection. So finding it in the throat should prompt a thorough search for infection at other sites, as the genital site is usually the primary source. For diagnosis, it's crucial that swabs from the pharynx be plated directly onto gonococcal selected media or tested using natties. This is because, as we mentioned, a presumptive diagnosis of gonorrhea cannot be made solely on the basis of seeing gram-negative Diplococci in smears from the pharynx.
Because of the normal throat bacteria.
Exactly. Other Neisseria species, notably Neisseria meningitidis, are common components of the normal flora in the throat and can appear identical under the microscope. Therefore, more specific tests like culture or nucleic acid amplification tests are absolutely required for pharyngeal sites to avoid misdiagnosis, and critically, for professionals thinking about the bigger picture of resistance. Because commensal or pharyngeal Neisseria are often naturally resistant to various antimicrobials, horizontal gene transfer, the sharing of genetic material between these harmless resident organisms, and gonorrhea within the pharynx may be a very important mechanism in the ongoing development of antimicrobial resistance in gonorrhea.
So the throat could be a mixing ground for resistance genes.
Precisely. It essentially makes the pharynx a potential resistance factory or reservoir for the pathogen, a key area of concern for drug development and understanding how resistance spreads globally.
How does gonorrhea present in the eyes of adults, and what makes it such a dangerous manifestation that requires immediate, urgent attention?
Ocular gonorrhea in an adult typically results from autoinoculation of gonorrhoeae from an infected genital site. So essentially, the individual touches an infected area like the genitals and then touches their eye, transferring the bacteria directly. Similar to genital infections, the manifestations can actually range quite widely, from severe, rapidly progressing disease to occasionally mild or even asymptomatic presentation. The variability in clinical manifestations may be attributable to differences in the ability of the specific infecting strain to elicit a strong inflammatory response in the eye tissues.
But when it's severe, it's really bad.
When it's severe, it's an ophthalmic emergency. The infection can result in a markedly swollen eyelid, severe hyperemia, which is intense redness, and chemosis, dramatic swelling of the conjunctiva, accompanied by a profuse, thick, purulent discharge. The massively inflamed conjunctiva can even drape over the cornea and limbus, the border between the cornea and the white part of the eye, sclera. What makes this so incredibly dangerous and why immediate professional intervention by an ophthalmologist is paramount is that lytic enzymes released from the infiltrating PMNs, those inflammatory cells rushing to the site, can cause corneal ulceration and, rarely, even perforation of the cornea itself.
A hole in the cornea.
Yes, leading to severe vision impairment or permanent blindness if not treated swiftly and aggressively. Therefore, prompt recognition and immediate treatment of this condition are of absolute paramount importance to preserve vision. The diagnosis is established by Graham's stain and culture of the purulent eye discharge. It's also crucial that genital cultures are performed concurrently, as the genitals are almost certainly the primary source of the infection, and treating only the eye would miss the underlying reservoir, risking recurrence or transmission.
This raises an important question, particularly for those working in pediatrics or obstetrics. What are the unique and truly serious implications of gonorrhea for particularly vulnerable populations, like pregnant women, newborns, and young children? The consequences here can be absolutely tragic if not managed properly.
For pregnant women, gonorrhea can have serious consequences for both the mother and the developing infant, which makes early detection during prenatal care absolutely critical. Early recognition of gonorrhea during pregnancy is crucial because it also identifies a population at high risk for other sexually transmitted infections, particularly chlamydial infection, syphilis, and trichomoniasis, requiring comprehensive screening to protect both mother and baby. The risks of salpingitis, inflammation of the fallopian tubes, and pelvic inflammatory disease, or PID conditions, which are associated with a high rate of fetal loss, are actually highest during the first trimester of pregnancy. Pharyngeal infection, which is often asymptomatic, may actually be more common during pregnancy, possibly due to altered sexual practices during this time, adding another layer of complexity to screening protocols. At term, common complications of untreated or undetected maternal gonococcal infection include prolonged rupture of the membranes, premature delivery, chorioamnionitis, inflammation of the fetal membrane surrounding the baby, pneumocystis, infection of the umbilical cord stump after birth, and even sepsis in the infant.
In these cases, N-gonorrhoeae can sometimes be detected in the newborn's gastric aspirate collected during delivery, indicating direct exposure to infected secretions. It's also important for professionals to remember that other conditions in microorganisms, including Mycoplasma hominis, Mycoplasma genitalium, Uriplasma urolithicum, C. trachomatis, and bacterial vaginosis, often accompanied by infection with Trichomonas vaginalis, have also been strongly associated with preterm birth and low birth weight. This highlights the interconnectedness of these maternal infections and the importance of a holistic approach to prenatal care and maternal health. Moving to neonates, the most common manifestation of gonococcal infection in newborns is ophthalmia neonatorum, severe conjunctivitis resulting from direct exposure to infected cervical secretions during parturition or childbirth. It's important to precisely distinguish prevention from treatment here.
Ocular neonatal instillation of a prophylactic agent, such as 1% silver nitrate eye drops, historically used, or ophthalmic preparations containing erythromycin or tetracycline, is effective at preventing ophthalmia neonatorum if administered shortly after birth as routine care.
But it doesn't treat it if it's already there.
Exactly. These prophylactic measures are not effective for its treatment once the infection is established. Treatment requires systemic antibiotics to truly eradicate the infection throughout the infant's body, not just eye drops. The clinical manifestations of ophthalmia neonatorum are acute and typically begin 2 to 5 days after birth. An initial nonspecific conjunctivitis with a serosanguineous, blood-tinged watery discharge is usually followed rapidly by tense edema of the eyelids, dramatic chemosis, swelling of the conjunctiva, and a profuse, thick, purulent discharge. The severe consequences of untreated ophthalmia neonatorum are devastating and include corneal ulcerations that can lead to nebulae, which are permanent clouding of the cornea or even perforation. This can then progress to anterior synechiae, adhesions within the eye, anterior staphyloma, bulging of the cornea, panophthalmitis, inflammation of the entire eye, and ultimately permanent blindness.
Truly devastating potential.
Infections described at other mucosal sites in infants, including vaginitis, rhinitis, nasal infection, and anorectal infection, are likely to be asymptomatic, making them harder to detect. Pharyngeal colonization has been demonstrated in about 35% of infants who also have gonococcal ophthalmia, and coughing is often the most prominent symptom in these cases, indicating respiratory involvement. The most common manifestation of systemic infection, or DGI, in the newborn is septic arthritis. The onset usually comes at 3 to 21 days of age, and polyarticular involvement affecting multiple joints simultaneously is common in neonates. Sepsis, meningitis, and pneumonia are seen in rare but extremely serious instances of neonatal DGI. For children beyond the neonatal period, it is a critical point that any sexually transmitted infection discovered raises the serious possibility of sexual abuse, necessitating a thorough, sensitive, and legally appropriate investigation. Gonococcal vulvovaginitis is the most common manifestation of gonococcal infection in children beyond infancy.
Anorectal and pharyngeal infections are also common in these children and are frequently asymptomatic, again making detection difficult without specific testing prompted by suspicion. The urethra, Bartholin's, and Skene's glands, and the upper genital tract are rarely involved in this age group, which helps narrow the diagnostic focus somewhat. Critically, all children diagnosed with gonococcal infection should also be evaluated comprehensively for chlamydial infection, syphilis, and possibly HIV infection, given the co-occurrence risks and the implications of potential abuse, emphasizing a comprehensive approach to child protection and health.
That's a sobering reminder of the infection's wide-ranging impact across all age groups. Now, here's where it gets particularly important for any professional aiming to truly understand the full scope of this disease, delving into the most severe end of the spectrum of gonococcal infection. Disseminated gonococcal infection, or DGI, often referred to simply as gonococcal arthritis, though it involves more than just joints. This is when the infection spreads beyond the initial local site, entering the bloodstream and causing systemic complications.
DGI truly represents the systemic reach of Gonorrhoeae, a powerful testament to its invasive potential when conditions are right. It results from gonococcal bacteremia, meaning the bacteria are actively present and multiplying in the bloodstream. Back in the 1970s, DGI occurred in approximately 0.5% to 3% of individuals with untreated gonococcal mucosal infection. The lower incidence of DGI we see at present is probably attributable to a decline in the prevalence of specific strains that are inherently more prone to dissemination, perhaps due to improved surveillance and earlier treatment eliminating those strains from circulation more effectively. Nevertheless, sporadic outbreaks of DGI still occur in places like North America, so it absolutely remains a significant concern, requiring a high index of suspicion in patients presenting with suggestive symptoms like arthritis or skin lesions. As we discussed earlier when talking about pathogenesis, DGI strains possess specific characteristics that seem to enable them to spread systemically more easily. They often resist the bactericidal action of normal human serum, meaning our blood's natural complement defenses struggle to kill them effectively. And crucially, they generally do not incite a significant local inflammatory response at the initial genital sites, probably because they generate limited chemotactic factors, the signals that call in our immune cells.
So the body doesn't notice them locally as much.
Exactly. This often translates to asymptomatic or very mild genital infections, allowing the bacteria to disseminate silently without alerting the host to the initial infection until systemic symptoms appear. Historically, strains recovered from DGI cases back in the 1970s were often of the poor but 1A serotype, were typically highly susceptible to penicillin, interestingly, and had special, more demanding growth requirements, including needing arginine, hypoxanthine, and uracil that made the organism more fastidious and actually more difficult to isolate in the lab back then, complicating earlier diagnostic efforts. For elite professionals, understanding the key risk factors for dissemination is critical for patient assessment and identifying those at higher risk. Menstruation is a major risk factor. Approximately 2/3 of DGI cases occur in women, and about half of affected women experience the onset of DGI symptoms within 7 days of the start of your menses. This strongly suggests that hormonal and physiological changes during menstruation may somehow facilitate bacterial spread from the cervix into the bloodstream.
Interesting connection.
Complement deficiencies, particularly involving the components responsible for forming the membrane attack complex, specifically C5 through C9, profoundly predispose individuals to Neisseria bacteremia in general. Therefore, persons who experience more than one episode of DGI should be carefully screened with an assay for total hemolytic complement activity, like a CH50 test, to identify this potential underlying genetic susceptibility. Additionally, DGI has been clearly associated with the use of the complement C5 blocking monoclonal antibody, acaluzumab, which is used for certain rare diseases. This association further highlights the critical role of complement in containing these infections, and serves as a cautionary tale for the potential infectious risks of targeted immune suppression therapies. The clinical manifestations of DGI have sometimes been classified into two stages, though it's important to remember that a clear-cut progression isn't always evident, and patients often present with overlapping symptoms. The first is sometimes called a bacterianic stage, which actually seems less common today. Patients in this stage typically have higher temperatures, fevers.
Polyarthralgias, meaning pain in multiple joints, are common and often occur along with tenosynovitis, which is inflammation of the tendon sheaths, particularly noticeable in the wrists, fingers, hands, and ankles, making specific movements quite painful. This often occurs concurrently with characteristic skin lesions.
Okay, so fever, joint pain, tendon pain, and skin lesions.
Yeah, that's the classic arthritis dermatitis syndrome picture. The polyarthralgias usually involve the knees, elbows, and more distal joints like the wrists, ankles, fingers, and toes, while the axial skeleton, such as the spine and pelvis, is generally spared, which can be a helpful diagnostic clue. The skin lesions are seen in approximately 75% of DGI patients and typically include papules, small raised bumps and pustules, pus-filled bumps, often with a distinctive hemorrhagic or bloody component at the base or center. Other non-infectious dermatitis manifestations like nodular lesions, urticaria, hives, or erythema multiforme have also been described, demonstrating a wide potential dermatologic spectrum. These lesions are usually found predominantly on the extremities, arms and legs, and typically number between 5 and 40, offering a clear visual diagnostic clue if present. For a clearer picture, referring to figure 156-2 in your source, you can visualize characteristic appearances like a very early petechia on a finger, an early papular lesion on the lower leg, a pustule with a central eschar resulting from an early petechial lesion, a mature lesion with central necrosis, black in center, on a hemorrhagic base, and even bullae or blisters on the anterior tibial surface.
So quite a variety of skin signs.
Yes. The differential diagnosis for this bacteremic stage of DGI is broad. It includes conditions like reactive arthritis, formerly Reiter syndrome, acute rheumatoid arthritis, sarcoidosis, erythema nodosum, drug-induced arthritis, and certain viral infections such as hepatitis B and acute HIV infection. It's important to note that the distribution of joint symptoms in reactive arthritis, for example, differs somewhat from that typically seen in DGI, as do the specific skin and genital manifestations, as illustrated comparatively in figure 156-3, which can help differentiate. The second stage, or presentation, is the joint localized stage with suppurative arthritis. This typically involves frank infection within one or two joints, oligoarthritis, most often the knees, wrists, ankles, and elbows, in decreasing order of frequency, though other joints are occasionally involved. Most patients who develop gonococcal septic arthritis actually do so without having had the prior polyarthralgias or skin lesions of the bacteremic stage.
In the absence of symptomatic genital infection, this particular presentation, acute arthritis in a young sexually active adult, cannot be reliably distinguished clinically from septic arthritis caused by other pathogens, like Staphylococcus aureus, requiring swift joint fluid aspiration and laboratory confirmation. The differential diagnosis of acute arthritis in young adults is indeed quite broad. In rare instances, osteomyelitis, a direct infection of the bone, can complicate septic arthritis, particularly when it affects the small joints of the hand, leading to potentially destructive outcomes if not treated aggressively. Finally, there are rare but extremely severe DGI complications that, while infrequent now thanks to antibiotics, demand immediate recognition and aggressive treatment. Gonococcal endocarditis, an infection of the heart valves, although rare today, was a relatively common and often fatal complication of DGI in the pre-antibiotic era, accounting for about a quarter of all reported cases of endocarditis back then. This highlights its historical severity. Another unusual but critical complication of DGI is meningitis, or inflammation of the brain and spinal cord membranes, which is always a medical emergency requiring immediate hospitalization and high-dose antibiotics.
Let's connect this now to an even broader public health context, one where gonorrhea plays a significant and deeply concerning role in transmission dynamics. Its critical interplay with HIV.
The association between gonorrhea and the acquisition and transmission of HIV has been clearly demonstrated in several well-controlled epidemiological studies, notably conducted in places like Kenya and Zaire, where both infections were highly prevalent. This is not just a theoretical correlation, but a documented causal increase in risk.
How does it increase risk?
Non-ulcerative STIs, like gonorrhea and chlamydia, are known to enhance the transmission of HIV by 3 to 5-fold. This heightened risk is thought to be due to several biological factors. Primarily, the inflammation caused by gonorrhea brings a high concentration of HIV-infected immune cells, like CD4 plus T cells and macrophages, to the genital tract, increasing the likelihood of transmitting HIV during sex.
Also, the inflammation leads to increased HIV viral shedding by persons who are co-infected with HIV and gonorrhea, meaning there is simply more virus present in their genital secretions.
So more virus shed and more target cells present.
Exactly. For example, HIV has been detected by PCR much more commonly in ejaculates from HIV-positive men who also have gonococcal urethritis compared to those HIV-positive men without urethritis. Furthermore, studies have shown that the HIV viral load in semen drops significantly by about twofold after appropriate antibiotic therapy for the urethritis. This emphasizes the profound impact that promptly treating the STI can have on reducing HIV transmission potential. And it's truly a two-way street, a dangerous synergistic vulnerability. Not only does gonorrhea enhance the transmission of HIV from an infected person, but it may also significantly increase an uninfected individual's risk for acquiring HIV in the first place if they are exposed.
How does it increase the risk of getting HIV?
A proposed mechanism for this increased acquisition risk is the recruitment of significantly greater numbers of CD4 plus T lymphocytes and dendritic cells, which are the primary target cells for HIV infection to the genital mucosa due to the inflammation caused by gonorrhea. These cells are essentially being drawn to the site of the gonococcal infection, creating a much more receptive and vulnerable environment for HIV to establish infection if exposure occurs. This effect seems particularly pronounced with non-ulcerative STIs like gonorrhea compared to ulcerative STIs. This highlights a critical and concerning synergy between these infections, where having gonorrhea can significantly fuel both the spread and acquisition of HIV, making dual screening, prevention, and prompt treatment absolutely paramount for public health. Okay, with that comprehensive understanding of the pathogen itself, its clever tricks, and its varied clinical impacts, let's unpack how elite professionals actually identify and confirm these infections in the lab. This diagnostic precision is absolutely crucial for selecting appropriate treatment, preventing complications, and enabling effective public health control measures like contact tracing. Absolutely. For men presenting with symptoms, a rapid presumptive diagnosis of gonococcal infection may often be obtained right in the clinic using Gram staining of urethral exudates. As we discussed, and as you can visualize in Figure 156-1 of our source material, the detection of those characteristic gram-negative intracellular monococci and diplococci, the coffee bean-shaped bacteria seen inside the patient's white blood cells, PMNs, is usually highly specific and sensitive for diagnosing gonococcal urethritis in symptomatic males. This quick result can be invaluable for guiding initial empirical treatment, allowing for swift intervention while awaiting more definitive tests.
But you emphasized earlier it's not so straightforward for women.
That's correct. The situation is much more challenging and requires a different approach for women. Gram stain is only approximately 50% sensitive in diagnosing gonococcal cervicitis.
Right.
That means it misses about half the cases, making it fundamentally unreliable as a sole diagnostic tool for ruling out infection in women. While PMNs, or white blood cells, are often seen in the endocervix on a Gram stain, and an abnormally increased number, specifically 30 or more PMNs per high-power field, does indicate the presence of an inflammatory discharge, cervicitis. The mere presence or absence of visible gram-negative intracellular diplococci in cervical smears does not accurately predict which patients actually have gonorrhea.
So you can have inflammation but not see the bacteria on the stain.
Exactly. Therefore, in this setting, the diagnosis in women should always be made by culture or another suitable non-culture diagnostic method like NATs. The sensitivity of a single endocervical culture, for instance, is around 80–90%, which is much better than Gram stain, but still not perfect, emphasizing the need for robust laboratory methods, especially the highly sensitive NATs. When it comes to sample collection and processing, certain steps are fundamental for ensuring accurate results, whichever test is used. Samples should ideally be collected using Dacron or rayon swabs, as cotton swabs contain fatty acids that can actually inhibit the growth of gonococci in culture. A crucial step, especially if culture is intended, is that part of the collected sample should be immediately inoculated onto a plate of modified Bayer, Martin, or other gonococcal selective media. Why immediately? It is absolutely vital to process all samples immediately, or get them into appropriate transport media quickly, because gonococci are very fragile and do not tolerate drying well. They will quickly die if left on a dry swab, leading to false negative culture results. If plates cannot be incubated immediately, they can be safely held for several hours at room temperature in candle extinction jars, which provide a low oxygen, high CO2 environment that helps keep them alive prior to incubation. For longer holding periods, for example, when specimens need to be mailed to a central lab, specialized culture media with self-contained CO2 generating systems, such as the Gembec or Gonopac systems, can be used to maintain bacterial viability during transport. And, as a best practice, it is always important to concurrently obtain specimens for the diagnosis of chlamydial infection, given the high frequency of co-occurrence, ensuring comprehensive patient care. There are also important site-specific diagnostic nuances that professionals must consider. For rectal infection, if a history of rectal sex is elicited, a rectal wall swab should be obtained, being careful to avoid contamination with feces, and then cultured or tested by gnats. For pharyngeal infection, remember, a presumptive diagnosis of gonorrhea cannot be made on the basis of seeing gram-negative diplococci in smears from the pharynx.
Because of the look-alike bacteria normally there?
Exactly. Other Neisseria species are common components of the normal flora in the throat and can appear identical under the microscope. Therefore, more specific tests like culture or nucleic acid amplification tests are absolutely required for pharyngeal sites to avoid misdiagnosis and ensure appropriate treatment, especially given the challenges of treating pharyngeal gonorrhea.
This sounds like a lot of intricate lab work, especially with culture. Here's where molecular technology, specifically gnats, has truly revolutionized diagnosis, hasn't it? Making it faster, often more sensitive, and less invasive. What are the key advantages and considerations here for professionals?
Indeed, nucleic acid amplification tests, or NOTs, have been a genuine game changer in STI diagnostics. Several NOT platforms, including the Roche-Covas Amplicore, GenProbe Aptima Combo 2, and BD ProbeTech ET, are now widely available on semi-automated or fully automated systems in most clinical labs. These tests work by detecting specific genetic sequences of N. gonorrhoeae and C. trachomatis. Generally speaking, NOTs are more sensitive than traditional culture for identifying both organisms, meaning they pick up more infections, especially low-level ones.
And the sample types are more flexible.
Yes, a major advantage, particularly with tests like the GenProbe and BD platforms, is that urine samples can be tested with a sensitivity similar to or even greater than that obtained when urethral swabs for men or cervical swabs for women are assessed by other non-NATs or culture, respectively. This makes screening much less invasive, particularly for men, and far more acceptable to patients, which leads to higher testing rates and improved public health outcomes through earlier detection. We're also seeing the emergence of cutting-edge point-of-care diagnostics. For instance, a point-of-care NAT-based test called Binx-IO, designed for both gonorrhea and chlamydia, is now approved by the U.S. Food and Drug Administration. It offers a remarkably rapid 30-minute turnaround time right in the clinic or doctor's office.
30 minutes? That's amazing.
It is. It allows for immediate treatment decisions and much faster partner notification, potentially interrupting transmission much more quickly. However, for elite professionals, there's a critical consideration, particularly when testing men who have sex with men, or MSM. It is absolutely essential to screen the rectum and pharynx in this population, not just the urethra or urine. Screening urine alone will miss the majority of infections in MSM, as these other sites are common reservoirs. This is a crucial point for effective public health strategy and individual patient care.
Are there any downsides to NAT?
Yes, a significant drawback of these powerful non-culture-based assays is that N. gonorrhoeae cannot be grown from the transport systems typically used for NAT samples. This means that if a culture is needed for confirmatory testing, rarely necessary now with validated NATs, or more importantly, for formal antimicrobial susceptibility testing, it cannot be performed from the original NAT sample. Susceptibility testing is vital in cases of suspected treatment failure or for public health surveillance in areas with high local resistance. This is a trade-off for the speed and sensitivity of NATs, and it necessitates collecting a separate sample specifically for culture if resistance testing is paramount. Now, when it comes to the diagnosis of gonococcal infection in children, there are extremely important legal implications, especially in cases of suspected sexual abuse, which require utmost diagnostic certainty. For this reason, the preferred method for diagnosis in children remains a standardized culture. However, regulatory bodies recognize the sensitivity of NATs, and two positive NATs, each targeting a different nucleic acid sequence from the organism, may be substituted for culture of the cervix or the urethra as acceptable legal evidence of infection in children.
What about rectal or pharyngeal testing in kids?
Although non-culture tests for gonococcal infection have not been specifically approved by the FDA for use with specimens obtained from the pharynx and rectum of infected children, NATs from these sites are generally preferred for diagnostic evaluation in adult victims of suspected sexual abuse, especially if the specific NATs have been evaluated by the local laboratory and found to be superior to culture. For children requiring testing, cultures should ideally be obtained from the pharynx and anus of both girls and boys, from the urethra of boys, and from the vagina of girls. Importantly,
Cervical specimens are generally not recommended for prepubertal girls due to anatomical reasons. For boys with a urethral discharge, a simple needle specimen of the discharge is usually adequate for culture. To ensure absolute accuracy and legal defensibility in these sensitive cases, presumptive colonies of N. gonorrhoeae grown from any culture should be identified definitively by at least two independent confirmatory methods: biochemical tests, antigen detection.
Finally, for the precise diagnosis of disseminated gonococcal infection, DGI, blood cultures are absolutely necessary in suspected cases, although they are positive in less than half of cases. The use of specialized isolator blood culture tubes may enhance the yield slightly, improving the chances of recovering the bacteria from blood. However, the probability of obtaining positive blood cultures decreases significantly after about 48 hours of illness, so prompt collection upon suspicion is vital. Synovial fluid aspirated from affected joints should be inoculated into blood culture broth medium and also plated directly onto chocolate agar, a non-selective enriched medium, rather than selected medium, because this fluid is unlikely to be contaminated with other commensal bacteria.
Is it easier to find in the joint fluid?
Gonococci are infrequently recovered from early joint effusions containing fewer than 20,000 white blood cells per microliter, but may be recovered more often from highly inflammatory effusions containing over 80,000 leukocytes per microliter. Interestingly, and somewhat diagnostically frustrating, the organisms are seldom recovered from both the blood and the synovial fluid of the same patient simultaneously. So you might find it in one site but not the other, making the diagnostic journey for DGI sometimes elusive and requiring careful clinical correlation with symptoms and other tests like skin lesion biopsies or mucosal site testing.
So, given all this intricate diagnostic information, all these nuances, what does this all mean for effectively treating these infections and, crucially, for combating that relentless rising tide of antibiotic resistance that we've discussed? This is really where the rubber meets the road for clinicians and public health.
This is truly the crux of the issue for any professional working in public health or clinical medicine today. Treatment failure for gonorrhea, which is becoming more common with rising resistance, can lead directly to continued transmission within the community. And even more critically, failed or inadequate treatment exerts selective pressure that fuels the emergence of further antibiotic resistance, creating a dangerous, escalating vicious cycle. The importance of providing adequate treatment with a regimen that the patient will actually adhere to cannot possibly be overemphasized. This is precisely why single-dose regimens have been extensively developed and are strongly preferred for uncomplicated gonococcal infections, to maximize patient compliance and ensure effective eradication of the bacteria, thereby limiting opportunities for resistance to develop or spread. The Centers for Disease Control and Prevention, the CDC, revised its treatment guidelines for gonococcal infections again in 2020, reflecting the latest resistance data. And these guidelines are critical for any professional in this field to know intimately. They are summarized in Table 156.1 in our source. The third-generation cephalosporin, ceftriaxone, given as an injection, is now recommended as the first-line regimen for uncomplicated gonococcal infection of the urethra, cervix, rectum, or pharynx. And importantly, the recommended dose has been doubled from previous guidelines to 500 milligrams intramuscularly, given as a single dose.
Why the dose increase?
This increase in dosage is a direct response to the observed doubling of the mean inhibitory concentrations, or MICs, of current circulating strains compared with the MICs seen just 20 years ago. We need higher levels to reliably kill the less susceptible strains. The ongoing development of decreased sensitivity, or MIC creep, to ceftriaxone throughout the world is a major global concern and will undoubtedly require the development of entirely new effective regimens in the near future, as our last reliable option is under threat. A significant change in the 2020 guidelines involves azithromycin. Azithromycin, which had previously been co-administered with ceftriaxone to provide additional treatment for gonorrhea and also to empirically treat potential coexisting chlamydial infection, is no longer routinely recommended as part of a first-line regimen for gonorrhea treatment alone.
So no more dual therapy as standard.
Not as the standard first-line approach for gonorrhea itself. This change was necessitated because resistance to azithromycin among U.S. isolates of N. gonorrhoeae, which had been very low, less than 0.6% for many years, increased dramatically, more than sevenfold to 4.6% in the most recent reported surveillance year. This rapid rise in resistance makes it unsuitable as a primary or synergistic agent for gonorrhea in many areas. However, if chlamydial infection cannot be excluded at the time of diagnosis, for example, if chlamydia testing wasn't done or results aren't back, Then concurrent treatment with doxycycline, 100 milligrams orally twice a day for 7 days, is still recommended to ensure that potential co-infection is adequately addressed. It's important to note that these recommendations for uncomplicated gonorrhea apply equally to HIV-infected individuals, maintaining a consistent standard of care across populations. The good news for now is that the currently recommended regimen for the treatment of uncomplicated gonococcal infection, that single 500 milligram intramuscular dose of ceftriaxone for urethral, cervical, rectal, or pharyngeal infection, almost always results in an effective cure in most parts of the world. It provides a reliable treatment option, which is why preserving its effectiveness is so critical.
What about the drugs that have fallen out of favor?
Right. As we discussed, quinolone-containing regimens are no longer recommended in the United States as first-line treatment because of the widespread resistance, QRNG. Similarly, rising MICs of the oral cephalosporin cefixime globally have led the CDC to discontinue its recommendation as a first-line treatment for uncomplicated gonorrhea, pushing us further towards reliance on injectable ceftriaxone.
Are there any viable alternative regimens if ceftriaxone can't be used, say due to allergy or availability?
Yes, there are alternatives, though they come with caveats. Multi-center trials of treatment for uncomplicated gonorrhea in the United States have shown high efficacy, greater than or equal to 99.5% for two specific combination regimens. First, gemifloxacin, a fluoroquinolone, 320 milligrams single oral dose, plus azithromycin, a high dose of 2 grams, single oral dose, and second, gentamicin, an aminoglycoside, given as a single intramuscular dose of 240 milligrams, or 5 milligrams in individuals weighing 45 kilograms, plus azithromycin, again, 2 grams, single oral dose. Note, the azithromycin here is mainly for potential chlamydia and possibly some synergistic effect, but resistance is still a concern. Also, a novel single-agent regimen, zoliflodacin, an experimental agent, 2 or 3 can single oral dose, showed 96% efficacy in trials, offering hope for future new oral options. Spectinomycin has been used as an alternative agent for treating uncomplicated gonococcal infections in penicillin allergic persons outside the United States, but it is not currently available in the U.S. market. It's also important to remember its limited effectiveness for the treatment of pharyngeal infection, which significantly reduces its overall utility, especially in populations like MSM where pharyngeal infection is common. So it's a less ideal option, even if it were available. Now, regarding test of cure, TOC, which is crucial for verifying eradication, especially with alternative regimens or resistant infections, and for monitoring resistance trends. Generally speaking, persons with uncomplicated genital or rectal infections who receive the recommended first-line regimen, ceftriaxone, do not need a routine test of cure.
Because it works so well.
Exactly. The cure rates are extremely high with the recommended ceftriaxone dose, and performing routine TOC isn't considered resource efficient or clinically necessary for uncomplicated cases. However, cultures for N-gonorrhoeae should be performed if symptoms persist after therapy with an established regimen. In such cases of suspected treatment failure, any Gonococci isolated must be tested for antimicrobial susceptibility to identify the resistance pattern. This is a critical public health surveillance action to detect emerging resistance. A test of cure is required, however, for pharyngeal infection regardless of the treatment regimen used. This should be done 7-14 days after treatment completion.
Why always for the throat?
Because symptomatic gonococcal pharyngitis is notoriously more difficult to eradicate pharmacologically than genital infection, due to poor antibiotic penetration into pharyngeal tissues. Therefore, TOC is needed to ensure eradication or to detect a possible treatment failure early. Asymptomatic pharyngeal infection can also serve as a persistent reservoir for transmission and resistance development, if not fully cleared. For individuals who cannot tolerate cephalosporins, e.g. due to severe allergy, and are treated with an alternative regimen like spectinomycin, if available, which has a particularly poor cure rate, 52% for pharyngeal infection, a subsequent pharyngeal sample should be cultured early, typically 3 to 5 days following treatment, as a test of cure, given the high risk of failure at that site. A single 2-gram dose of azithromycin might be considered if the infecting organism is known to be sensitive based on local surveillance data or susceptibility testing, or in areas where rates of resistance to azithromycin remain documented as low. Similarly, quinolones may only be used if the infecting organism is definitively known to be sensitive through testing. If culture is not readily available and innate remains positive after treatment, every effort should be made to perform a confirmatory culture to enable resistance testing. And critically, all isolates recovered from any test of cure cultures should undergo full antimicrobial susceptibility testing to monitor for resistance and inform public health strategies.
And what about reinfection?
That's a huge issue. Because of the documented high rates of reinfection with N-gonorrhoea, and often C. trachomatis as well, occurring within 6-12 months after initial treatment. Persons previously treated for gonorrhea should be retested approximately 3 months after their treatment, regardless of whether they have symptoms or believe they were re-exposed. This emphasizes the need for ongoing vigilance, risk reduction counseling, and repeat screening in high-risk individuals. When it comes to treatment for specific, more severe conditions, the management strategies differ. Treatments for gonococcal epidemiolitis and pelvic inflammatory disease, PID, are complex and typically involve longer courses of combination therapy. They're discussed in detail in Chapter 136 of our source material, indicating their specialized management needs. For ocular gonococcal infections in older children and adults, the recommended management involves a single, higher dose of ceftriaxone, 1 gram intramuscularly, combined with immediate and copious saline irrigation of the conjunctivae. Both interventions should be undertaken expeditiously.
Additionally, patients must undergo a careful ophthalmologic evaluation, including a slit lamp examination, to assess for corneal damage and guide further management, emphasizing the urgency of preserving vision in these cases. For disseminated gonococcal infection, DGI, particularly the joint localized stage with suppurative arthritis, higher dosages and longer durations of antibiotic therapy are typically required, as detailed in Table 156-1 in your source. This often involves an initial period of intravenous therapy, followed by oral therapy, reflecting the seriousness of systemic infection. Hospitalization is generally indicated if the diagnosis is uncertain, if the patient has localized suppurative arthritis that requires repeated aspiration of joint fluid for drainage and diagnosis, or if the patient cannot be relied upon to comply with a complex outpatient treatment regimen. Open surgical drainage of an infected joint is necessary only occasionally, for example, for managing infections in the hip, which can be difficult to drain adequately percutaneously. Non-steroidal anti-inflammatory agents, NSAIDs, may be indicated alongside antibiotics to alleviate pain and hasten the clinical improvement of affected joints. Finally, gonococcal meningitis and endocarditis, being life-threatening complications, should always be treated aggressively in the hospital with high-dose intravenous ceftriaxone, typically 1 to 2 grams intravenously every 12 to 24 hours. Therapy needs to be prolonged, usually for 10 to 14 days for meningitis and for at least 4 weeks for endocarditis, reflecting the severity and difficulty of eradicating these deep-seated infections. And it's absolutely crucial that all persons who experience more than one episode of DGI be thoroughly evaluated for an underlying complement deficiency, as this genetic predisposition significantly increases their risk for recurrent systemic Neisseria infections. And identifying it is key to counseling and potentially preventing future life-threatening episodes.
That detailed look at diagnosis and treatment really shows the complexity involved, especially with resistance complicating things. But ultimately, for elite professionals and for public health in general, the long-term solution has to lie in prevention. So, as we wrap up our deep dive today, let's consider how we prevent this infection in the first place and limit its spread in the broader community. Given the immense challenges posed by antibiotic resistance, prevention becomes even more paramount, doesn't it?
Absolutely. Behavioral prevention, as you might expect, plays a significant and often primary role in individual protection. Condoms, if used properly and consistently during any type of sexual activity, provide effective protection against both the transmission and acquisition of gonorrhea, as well as other infections that are transmitted primarily to and from genital mucosal surfaces. This remains a fundamental cornerstone of individual prevention messages.
What about spermicides?
Well, spermicidal preparations used with the diaphragm, or cervical sponges impregnated with the spermicide nonoxynol 9, do offer some degree of protection against both gonorrhea and chlamydial infection. However, this is where a nuanced understanding for professionals is absolutely vital, because a seemingly protective measure also carries a potential risk, especially concerning HIV. The critical caveat with nonoxynol 9 needs to be understood. The frequent use of preparations that contain nonoxynol 9 is actually associated with mucosal disruption. This means it can irritate and cause microscopic damage to the delicate lining of the vagina or rectum. And that mucosal disruption paradoxically may enhance the risk of HIV infection in the event of exposure to HIV.
Because it creates entry points for the virus.
Exactly. It compromises the natural barrier. This highlights the complex interplay of prevention strategies and the critical need for evidence-based recommendations, moving away from nonoxynol 9 for STI/HIV prevention specifically. Beyond individual behavior, comprehensive public health strategies are absolutely crucial for controlling gonorrhea at the population level. A key component is partner notification and treatment. All patients diagnosed with gonorrhea should be carefully counseled and strongly encouraged to refer their sex partners for evaluation and treatment. This referral is particularly important if their last sexual contact with the partner took place within 60 days before the onset of their own symptoms or the date of their diagnosis. If the patient's last potential sexual exposure was more than 60 days prior, then their most recent sex partner should still be evaluated and treated.
Getting partners treated is key to stopping reinfection.
Precisely. And to facilitate this, another innovative strategy gaining traction is partner-delivered medications or prescriptions, sometimes called expedited partner therapy, EPT. Where not prohibited by state laws or regulations, this approach allows a healthcare provider to give the index patient medication or a prescription to deliver directly to their partner without the partner needing a separate medical evaluation first. This significantly diminishes the likelihood of reinfection or relapse due to treatment failure in the partner, in the index patient, and helps treat partners who might otherwise face barriers to accessing care. It's an important option for partner management aimed at breaking chains of transmission more efficiently. Patients should also, of course, be instructed to abstain from sexual intercourse until their own therapy is completed and until both they and their sex partners no longer have any symptoms.
So, what does this all mean for broader public health education and screening efforts, and how does it fit into the bigger picture of STI control and overall global health strategy?
It means there must be a continued and even greater emphasis on prevention through multiple channels. Comprehensive public health education campaigns, individualized patient counseling focused on risk reduction during clinical encounters, and ongoing efforts towards behavior modification, particularly promoting the consistent and correct use of condoms. Furthermore, sexually active persons, especially adolescents and young adults who bear the highest burden of infection, should be proactively offered routine screening for STIs, including gonorrhea and chlamydia,
Even if they don't have symptoms.
especially if they don't have symptoms, given the high rate of asymptomatic infection. For most male patients, as we discussed, a netta of urine, or a urethral swab, can be effectively and non-invasively used for screening. For women, cervical or vaginal swabs or urine for gnats are standard screening methods. Increasing screening uptake is critical for detecting and treating those silent infections that drive the epidemic. And we must always remember the broader impact. Preventing the spread of gonorrhea may significantly help reduce the transmission of HIV, underscoring how interconnected these public health battles truly are. Controlling one STI can have major cascading positive effects on others.
This has been an incredibly detailed and, at times, quite sobering look at a truly tenacious pathogen. But for any professional, and for all of us really, there's always the horizon to consider. Despite its long history and the relentless challenges it presents, particularly with resistance, is there an effective vaccine for gonorrhea finally on the horizon? Or is that still just a distant dream, given its incredible adaptability and immune evasion skills?
That's the multi-billion-dollar question, isn't it? Currently, unfortunately, no effective vaccine for gonorrhea is yet available for widespread use. This is largely due to the bacterium's remarkable antigenic variation, its ability to constantly change its surface proteins like PLE and OPA, and its sophisticated immune evasion strategies, like mimicking host sugars with LOS sialation or producing blocking antibodies via REAM, which we discussed earlier. These features make it an incredibly difficult target for a conventional vaccine approach that relies on stable antigens.
So the usual vaccine strategies don't work well.
They haven't so far. However, efforts to develop and test several novel candidate vaccines are indeed underway, leveraging new technologies and understanding of gonococcal biology. These efforts offer a glimmer of hope for a more robust and sustainable prevention strategy in the future. A particularly promising and somewhat unexpected recent development highlighted in our source material is a field trial involving a licensed Group B meningococcal vaccine called Bexaro, 4C-MeMB.
A meningitis vaccine helped with gonorrhea.
Yes, intriguingly. This vaccine, which targets several outer membrane proteins of Neisseria meningitis, had previously been shown in its prototype form, used during an outbreak in New Zealand, to correlate with a reduction in the incidence of gonorrhea in the population that received the vaccine to control that specific Group B meningococcal epidemic. This observation has spurred further research.
So it may be cross-protection.
Exactly. It provides a fascinating and potentially exciting pathway for future gonorrhea vaccine development, suggesting that we might be able to leverage cross-protective immunity generated against shared or similar proteins found in other Neisseria species, like meningitis, to finally develop an effective gonorrhea vaccine. If this approach proves successful, it would be a true game changer in the fight against this resilient STI. #TagOutro.
And that wraps up our deep dive into the incredibly complex world of gonococcal infections, focusing intensely on Chapter 156. What we've really seen today is that gonorrhea is far, far from being a simple, easily dismissed infection. It's a persistent and constantly evolving public health challenge, marked by its incredibly complex microbiology, its dynamic and often alarming epidemiological shifts, its intricate mechanisms of pathogenesis and immune evasion, and most critically, perhaps, a relentless and ongoing rise in antibiotic resistance that threatens our ability to treat it. We've traced how its impact stretches from seemingly localized infections in the urethra or cervix to severe, potentially life-threatening disseminated disease affecting joints, skin, heart, and brain. We've seen how it affects diverse populations differently, and tragically, how it deeply intersects with other major public health crises like HIV.
Absolutely. And mastering this information like an elite professional, as we set out to do, means gaining a profound appreciation for the truly multifaceted nature of this infection. It's about understanding everything from its sophisticated molecular mechanisms of immune evasion, like the phase variation of LOS allowing it to change its sugar coat, or the cunning roles of porin and OPA proteins in manipulating our cells and immune responses, or even the paradoxical effect of RNA antibodies, all the way to the complex societal factors that influence its rapid spread and our ongoing difficult struggle to find consistently effective treatments and, with considerable hope, a preventive vaccine.
Right.
Truly understanding these intricate details, from the genetic basis of resistance to the nuances of clinical presentation and diagnostics, allows for genuinely informed decision-making in personal health, clinical practice, and a much deeper appreciation for the complex, often frustrating, but vital work involved in public health efforts worldwide to control this adaptable pathogen.
So as we close this deep dive, let's leave you with a provocative thought to mull over. Given Neisseria gonorrhoea's remarkable capacity for antigenic variation, its ability to generate blocking antibodies, and now this intriguing potential for cross-reactivity with other Neisseria species like meningitis, how might the strategies for developing a truly effective and long-lasting gonorrhea vaccine need to differ fundamentally from those used successfully for other pathogens? Will we need vaccines that target multiple, constantly changing antigens, or perhaps focus on conserved proteins, or even aim to modulate the immune response itself? What are the broader implications of finally achieving such a vaccine for the global fight against STIs? And what might be the next scientific or public health frontier in our ongoing centuries-long battle against this incredibly adaptable microbial foe? Thank you for joining us on this deep dive, and we look forward to continuing your learning journey with us next time.